So here we are at the second half of this lecture, Lecture 3, covering again Chapter 4, the structure of proteins considered in 3 dimensions. We already talked about the levels of protein structure, but in the last half of this lecture we talked about primary and secondary structure. We're going to pick up in just a moment right where we left off, and we'll talk about tertiary structure, followed by quaternary structure. And then, as promised, we'll speak very briefly about how proteins get to these structures and what makes them fold, and just a little bit about how we visualize proteins experimentally to determine their structures in the first place. So before we dive right into tertiary structures, let's again get some terminology under our belts here and talk about different styles or categories of protein conformations or shapes. We refer to these as fibrous versus globular. Fibrous proteins are named that because they very much resemble fibers. They tend to be long, very narrow, and they're extremely strong and stable. I think of fibrous proteins as resembling rope, made up of very long, slender, narrow fibers that are braided together to give a great deal of tensile stability. More importantly, fibrous proteins are static. They assume their shape, they stay in that shape, and they don't do nothing after that. Static proteins, fibrous proteins, aren't machines. They are not nano robots. They're not nano machines. They're not nano tools. They are just structural. They are rope. So we need these fibrous proteins. These are the proteins that hold our skin into our shape. They hold our organs in place. And they allow our cells to stretch and deform without tearing or fracturing. But they don't do anything dynamic. They don't change their environment. They don't accomplish a specific function or task. They are there to hold things together. So here's an example of a fibrous protein. This is collagen, the protein that makes up your hair and fingernails. Again, very structural, very static, and not dynamic. Really not all that interesting. I liken these to the beams and girders and studs of the buildings that we're still using as our analogy. Necessary? Absolutely. Can it stand without these structures? Absolutely not. Interesting? No. And that's what fibrous proteins are. Globular proteins are the ones that we already started talking about in the last lecture. These are the proteins that fold back on themselves, assume a very three-dimensional and kind of lumpy, pseudo-knotty shape. That shape is roughly spherical, which is where these get the name globular. And because these proteins are doubling back and folding within themselves, these amino acid chains are, again, doubling back and folding within themselves, we can have residues that are very far apart in the amino acid chain being very close together. And what I mean by that is if you imagine the amino acid chain, the primary structure, as a piece of string, imagine balling that string up into a three-dimensional shape, a three-dimensional ball, and you could see where pieces of the string that are very far apart along its length could be right up against each other and touching because you've balled it up and made it take this three-dimensional shape. Globular proteins folding into shapes like this allow catalytic sites to form. These are the active sites of the enzymes we will be introducing in just a few lectures. So we are now talking about our protein machines, proteins that do specific biochemical tasks in the cell. All of the nuts and bolts of what's going to happen in an enzyme occur at these catalytic active sites, and those sites form by those proteins folding in a particular shape. Oh, did I just hint at the structure of a protein allowing to do its catalytic job? Yes, I did. Proteins assuming their native conformation and folding into their specific shapes are what allow those enzymes to do their specific jobs biochemically. Structure equals function. So here is myoglobin. This is the protein that carries oxygen in your muscles. It offloads oxygen from the blood and carries oxygen within the muscles itself. Myoglobin's function is to carry oxygen. We see oxygen as part of a heme ring there in the center, and it is the three-dimensional shape or native conformation of myoglobin, all of these alpha helices turning with our turns, our proline glycine turns, turning amongst themselves to assume this three-dimensional shape that allows this myoglobin to do its job. I will come right off the bat here and admit to you that this is about to be a weak analogy, but we're going to come back to this analogy in a few lectures and it's going to be much more powerful. I want you to start thinking about enzymes and these functional proteins, these globular proteins, as a bread machine. This bread machine has a very specific function to make bread, And it can do that function because of all the disparate parts of the bread machine coming together in three-dimensional space in just the right way. We have the computer component of the bread machine there on the right. We have the basin or the loaf pan for the bread there in the center. For those of you who have had or seen a bread machine, there's a mixing arm in the bottom of that loaf pan. Obviously, there's an oven component to the bread machine that allows the bread to bake. So there are all these different parts that come together into this very unique and specific three-dimensional structure of the bread machine that then allows this bread machine to do its job. This is what we're getting at with globular proteins. Different secondary structures coming together in a very unique and specific way to create structures, larger structures within the protein that allows that protein to do its job. So we have scratched the surface now of the notion of tertiary structure. Tertiary structure, said simply, is the fully folded protein. But to get there, we have taken all of our secondary structures and all of the individual atoms of the single polypeptide chain and brought them together into the native conformation that makes the three-dimensional shape of that protein that we're after. So tertiary structures include the arrangement and three-dimensional position in space of all the side chains of those amino acids, all of the prosthetic groups, these are the things that the amino acids and proteins are holding onto, such as heme, in that case of myoglobin, and the arrangement of everything else, including all the turns and secondary structures with respect to each other. Again, to say it simply, the tertiary structure is the completely folded protein chain, but only for a single continuous polypeptide chain of amino acids. We're going to talk about what happens when multiple protein chains come together to form larger structures. That's quaternary structure. But for tertiary structure, we're talking about a single chain of amino acids from its amino terminal end to its carboxy terminal end, fully folded into its native conformation. That is your tertiary structure. Folding, again, like that example from the pieces of string, folding can bring amino acids that are very far apart in the chain, very close together, and allow them to interact. The side chains of these amino acids can then also interact in favorable ways. You can imagine if one side chain is negatively charged and another side chain is positively charged. If those side chains of those amino acids are brought close together in three-dimensional space, opposites attract, and that will stabilize the protein folding. So again, back to this example of a pile of string. A protein, a fully folded tertiary protein, tertiary structure protein, is very much like a pile of string, where the string itself is the chain of amino acids, but that pile of string is allowed to fold in a multitude of different orientations or conformations. Without forces holding that protein into the specific shape that we want, its specific native conformation, you can imagine that loose string is just going to fall into some other shape and not have the ability to do its job. The forces that stabilize the folding of a protein into its tertiary shape, the forces that allow a protein to assume its native conformation and then stay there, are typically non-covalent forces. Of course, these include hydrogen bonds. Hydrogen bonds between polar groups in the side chains of polar non-charged amino acids. Remember, if we're talking about hydrogen bonds between amino and carboxy groups of the peptide backbone itself, we're talking secondary structure stabilization. If we're going to have tertiary structure stabilization, those hydrogen bonds must solely be between the side chains, the variable groups. Other non-covalent forces that can stabilize native conformations are electrostatic interactions. The example I gave you just a moment ago, when a negative side chain interacts favorably with a positive side chain, keeping those amino acids together and keeping that protein in that tertiary shape. Remember, when we talk about negative and positive side chains, we should really be using other terms. It's positive side chains that are basic amino acids, and negative side chains are acidic amino acids. I want you to think about something for just a second now. We're talking about folding a protein into a three-dimensional shape, and we have to, in this course, constantly build and remain conscious of the relevancy of the material we've learned before. I'll say this for the first time now. I mentioned it in the very first day when we met and went over the syllabus, and I will say it more and more as we go down the course and continue to have these lectures together. But unlike many of the other courses you will take in your curriculum, biochemistry is not a collection of separate stories thrown together into a single course. It cannot be siloed, and what I mean by that is you can't forget about things that we've learned about previously in the course, even if we've already taken an exam on it. We will continuously and constantly build on concepts and topics from day one all the way until the end of this course. So with that in mind, I want you to think about acidic and basic side chains. Think about them folding into the native conformation of a larger protein, and ask yourself, where in the context of that fully folded protein would you expect those side chains to be? This is our first example of an exam type question. I'll encourage you to pause the lecture now and think about this, give it some real thought. Imagine that this was an exam question and think about how you would answer it. You already know everything you need to know in order to get this question right. I'm going to wait probably 3 seconds and then I'll answer it myself. So you would find these amino acids on the exterior face of the protein. Acidic and basic side chains are the most hydrophilic side chains of the 20 amino acids. They are fully charged, very hydrophilic. They interact extremely favorably with water. Never forget that we are bags of water, one of the first concepts on the first slides of this course. We are bags of water. Our cells are microscopic bags of water. These proteins are folding in a watery environment. So all of the hydrophobic amino acids that we discussed in those first lectures are racing towards the interior of the protein, trying to run and shield themselves away from water. Remember, those hydrophobic amino acids don't really care about one another. There's no real interaction between them at all. They are the US and Russia fighting against Nazi Germany in World War II. Their mutual enemy makes them friends. The hydrophilic amino acids, on the other hand, they race to take up positions on the exterior face of the protein where they can and will interact favorably with water. Positive and negative side chains are the most hydrophilic and so the most eager to interact with water, and so we would find charged side chains on the outside of a fully folded protein. Again, hydrophobic interactions are another non-covalent force that we see stabilizing tertiary structure. This is not a true interaction. These are perceived interactions, but they do stabilize tertiary structure. I mentioned it a moment ago, but we would find all hydrophobic amino acids largely on the interior of the protein, hiding from water and mutually excluding water. Covalent forces, too, sometimes contribute to the overall shape and stability of tertiary structure. Of course, peptide bonds are keeping the amino acids together, so it would be inappropriate to not mention them. The protein wouldn't be a protein without peptide bonds, so that goes without saying. Oops. But we've talked about other covalent bonds in the context of proteins that kind of supersede peptide bonds. The kind of bonds, covalent bonds, that can keep proteins into three-dimensional shapes. Do you remember what those are? Those were our disulfide bridges between cysteine residues. Disulfide bonds are covalent bonds that can hold two cysteines together, and correctly placed disulfide bonds can help stabilize tertiary structure and keep proteins folded into their proper shape by keeping different amino acids close to one another in physical shape. So here's an example of a disulfide bridge in general. And we also talked about disulfide bridges keeping antibodies, or immunoglobulins, in their correct three-dimensional shape, allowing them to do the job of binding to antigens. So this is from your textbook, but this is a really nice figure that captures everything we've been talking about, even into the last lecture chunk, in one visual. So we have here on the left some examples of secondary structure, native secondary structure. We have an alpha helix here, this tightly coiled chain of amino acids, stabilized by hydrogen bonds between the amino and carboxy groups themselves, those hydrogen bonds parallel to the axis of the alpha helix. We have a beta sheet made up of two beta strands. I know that this is an anti-parallel beta strand because this strand goes up, N to C, and it double backs, and this one comes down in the opposite direction. And here the hydrogen bonds holding those beta strands together are represented by these bars. We can also have stabilization by the coordination of a prosthetic group. Here, that coordination is a single ion, some metal ion. The metal ion is serving as a bridge, and we can see that it's interacting with amino acids on either side of it. This metal ion has a positive 2 charge. So what kind of amino acid do you think might be here? If you said an acidic amino acid, you're absolutely correct and give yourself 2 points for being awesome. Acidic amino acids are negatively charged. Negatively charged amino acids are going to interact favorably with a positively charged metal ion. So this negative acidic amino acid here and this negative acidic amino acid here are being brought close together in three-dimensional space and stabilized because they're interacting electrostatically with this positively charged metal ion. Here we see some hydrophobic interactions between our most hydrophobic aliphatic amino acids, isoleucine, valine, and leucine. They're hiding here, hiding away from water in the center of the protein, shielding themselves from water by mutually huddling together to exclude it. These 2 cysteines are being held close together in three-dimensional shape, and I know they're cysteines because what's holding them there is this disulfide bridge. Disulfide bridges are only possible between cysteine residues. We have here the example of hydrogen bonding, keeping 2 amino acids close together in three-dimensional space, and finally an electrostatic interaction between positive and negative groups here. So all of these non-covalent and covalent as well, all of these non-covalent and covalent forces are able to keep proteins held into their specific, functional, three-dimensional shape that we call the native conformation. And when proteins assume this shape, they are in their tertiary conformation. They are at the tertiary level of protein structure. Just as we said that not all proteins contain alpha helices or beta sheets, it's important to note that not every single protein uses all of the forces we're talking about here. You can have proteins that just use hydrophobic and H-bond interactions. You can have proteins that rely heavily on disulfide bridges and electrostatic interactions. But all of these forces can contribute to a stable tertiary shape. You're very likely to find hydrogen bonds, electrostatic interactions, and hydrophobic interactions in the vast majority of proteins. Those 3 are the most common. And it is the sum total of all of these forces together that stabilize the protein into that good, proper, native conformation that we want it to assume. And once it is in the right structure, once that protein has achieved the correct shape, it can then have the correct function and do its job. That brings us to quaternary structure, level 4, the last level of protein structure. Well, if tertiary structure is the fully folded protein, if we really have folded everything we possibly can after tertiary structure, then what are we doing with quaternary? Aren't we done? Like, what is there after the fully folded protein? Well, sometimes proteins will engage in interactions with other proteins and form larger structures. Some protein machines, some enzymes, are actually made up of more than one single polypeptide chain of amino acids. We call these types of proteins or enzymes complexes, the term coming from making things more complex or bringing many parts together. When you have a complex, by definition, you know you're dealing with a protein that's made up of more than one individual polypeptide chain, and then each one of those individual polypeptide chains is referred to as a subunit of the larger complex. So if you have a complex, by definition, you must have 2 or more subunits. Subunits of complexes could be identical proteins. They could be different proteins as well. Common complexes are referred to as dimers. That means there are 2 subunits. Trimers, 3 subunits. Tetramers, 4 subunits. And then we kind of stop with the quantifying of the number of subunits. And after that, we use the term oligomer. And oligomer means anywhere above 4 subunits. So this is an example of a single protein. This is myoglobin, once again on the left. A single protein. It is not a complex because there's only one polypeptide chain involved, and this protein folds into its tertiary shape, and it's done. There's no quaternary structure for myoglobin. It folds into the proper shape. It carries a heme group. It can carry oxygen in the muscle, and you're done. Hemoglobin, on the other hand, is here on the right, Hemoglobin is a complex. It's actually a tetramer. It's made up of 2 identical alpha subunits and 2 other identical beta subunits. 4 subunits in total, making it a tetramer. Each one of those subunits can carry a single heme prosthetic group, and each one of those hemes can carry an oxygen molecule. So hemoglobin can carry 4 oxygens, whereas myoglobin can only carry 1. Hemoglobin is a complex, myoglobin is not. Hemoglobin assumes quaternary structure, and myoglobin does not. And I said all that. So, if we have individual subunits interacting together to form a complex, they have to be able to interact. They have to stick to one another. Otherwise, they'll just fall apart from each other and float away. So, much like stabilizing tertiary structure, subunits interact with one another through non-covalent forces. The same non-covalent forces we've already discussed. Hydrogen bonds, hydrophobic interactions, induced dipole-dipole interactions, the van der Waals interactions we talked about a while ago, electrostatic interactions can be involved as well. And there are some covalent forces that can bring subunits together, most notably disulfide bridges, which would be between cysteines on different polypeptide chains in this case. Because the subunits within complexes are so intimately in contact and touching so much, a change, a conformational change, an induced shape change in one subunit can be transmitted or felt by another. It's a long ways away now. We're talking over 8 years ago. But when President Obama was first elected in 2008, Inauguration Day in Washington, D.C. in January of 2009 was huge. It was huge. In fact, it was this huge. So many people turned out to see this historic inauguration, and rightfully so, of course. These people were intimately in contact. They were shoulder to shoulder. The crowd was deep, and it was dense. Now I want you to imagine, this is hypothetical, it probably didn't happen, but I want you to imagine that someone in that crowd fainted. They overheated, they were overcome with emotion of the moment, and they fainted, they dropped. Of course, you would expect that people around the faintee, we'll call them, moved back to give them some room. Formed a ring around them, gave them a space, made sure they were okay, helped them up, made sure they could get air. With a crowd so crowded, as those people around the faintee stepped back, they jostled the people behind them and crowded them. And those people then stepped back and jostled those behind them. And they stepped back and jostled those behind them. And so now, next thing you know, is you have this wave of motion and jostling perpetuating across this entire crowd because of this one person fainting. You can be 50 yards away from this faintee. You don't even know someone fainted. And then all of a sudden you're being pushed and jostled out of the blue as a direct result of that wave of motion that's occurring 50 yards away. The same exact thing can happen in complexes. Because complexes are so intimately in contact with one another, a change in one subunit, a shape change that's induced by maybe another molecule binding or some disruption of an interaction, that change in shape can propagate like the jostling of people on Inauguration Day and move itself through the entire complex, even spanning subunit to subunit. Here's an example of what I mean. So this is also hemoglobin. We're looking at the 2 forms of hemoglobin, the oxy form where the light blue oxygen is in the active site, and the deoxified form where the oxygen is absent. When oxygen binds, it's almost like the person fainting. The side chains around the oxygen move to accommodate the oxygen binding there. Oxygen takes up some space, and so the side chains move as a result of that. As those side chains move, they jostle the side chains next to them. And those side chains jostle their neighboring side chains, just as the people in the crowd are jostling each other. And before you know it, you have these amino acids so far away from that active site, moving as a direct result of oxygen binding. They're moving because they've been jostled by the amino acids next to them. which were jostled by those next to them, which were jostled by those next to them, which, on and on, were jostled because of these amino acids in the active site being jostled by oxygen. Pretty incredible. We call this a conformational change, and these will play very large roles in enzyme activity as we go further down into this unit. Any protein that changes its shape as a direct result of being bound by something can also be referred to as allosteric. Allosteric literally translates from its Latin roots to mean other shapes. Allo means other, and steric means shape or space. So allosteric proteins are proteins that can change their shape in direct result of something happening to it. So let's review what we talked about in these two lecture chunks. The primary structure of a protein, the primary level of protein structure or folding, is the chain of amino acids itself, and nothing more. The chain of amino acids from the amino terminal end to the carboxy terminal end. Those amino acids can then begin to assume localized regions of order, such as alpha helices and beta strands. Beta strands can double back on themselves, forming larger beta sheets. These secondary structures, again, are quite small. They're a localized order only, and they're stabilized by hydrogen bonds within the amino acids themselves. Once these secondary structures has formed, those secondary structures can then come together to form larger regions of order, and that's what we call tertiary structure. Tertiary structure is the fully folded single polypeptide chain of amino acids taking on its native conformation. And let's not forget that if this amino acid is going to be, if this alpha helix is going to be connected to this one, if this beta strand is going to be connected to this one, we not only need helices and strands, we need turns as well. Some proteins, and I should say a large percentage of proteins, aren't done when they've reached their tertiary shape because those proteins are actually meant to be subunits of larger protein complexes. Quaternary structure is when individual polypeptide chains, fully folded, come together and interact with one another through non-covalent forces and disulfide bridges to form a larger protein complex. I know we're not doing clicker questions here, but I did leave these two clicker questions in place because I wanted you to have some sense of the context of everything we've talked about. This question is asking you what is indicated by the circle. What we mean by that is what's highlighted in red here. What is this showing? I'm going to answer it in just a second, but pause it if you need some time. What this is showing, highlighted in red, is, of course, a beta sheet. I said, of course, and then I stumbled over the answer. Beta sheet. That beta sheet is made up of 3 beta strands. We can get a little bit more technical and say they're anti-parallel because they're looping and switching back on themselves. But if we're talking beta sheet, we are talking secondary structure. So the correct answer to this clicker question is B. Let's compare that and contrast it to this question. What's being indicated by the circle now? Again, pause it if you need some time. I'll go ahead and answer it in a second. If we look at this picture, we see it's a single polypeptide chain. Here is its beginning. I know this is the beginning because I see the amino terminus, the N terminus here. Then we have an alpha helix connected to a beta strand, turn, making the full beta sheet. We connect to another alpha helix, a pretty sharp turn to the last alpha helix, and then we end at the carboxy terminus. A single polypeptide chain that has fully folded and brought all of those secondary structures together into one final shape. That is the tertiary structure of this protein. So the correct answer here would be C. A little bit about protein folding here. In fact, we've hit on this subtly already in this chunk of the lecture. In many ways, protein folding deserves its own course. In a graduate school, you can take a course in protein folding. We'll cover it in just a few minutes here that I have for this slide. We've already said that the reason a protein folds into its native conformation is dictated solely by its sequence. The amino acid sequence, its primary structure, is what is directly responsible for the protein folding into its tertiary shape, its native conformation. This idea of sequence dictates structure, and then structure dictates function. But even with that direct relationship, believe it or not, as biochemists, we still cannot predict the three-dimensional shape of a protein given only its amino acid. Textbook says that we can kind of do this. The textbook is wrong. We can't do this. We can guess, and sometimes we guess right. But don't poo-poo the fact that we are guessing and nothing more. So proteins fold into three-dimensional shapes using many, many factors. Many different things cause proteins to fold into three-dimensional shapes. And the reason why we can't do this prediction with a high degree of accuracy is because we don't yet understand all the factors that go into protein folding, but we do understand the basics. The largest contributor to protein folding is hydrophobicity, and this is what we mentioned earlier in this lecture series for Lecture 3. The need of hydrophobic nonpolar side chains rushing into the interior of a globular protein so that they can shield themselves from water and exclude water, while at the same time these polar hydrophilic side chains, remember, most notably the acidic and basic ones with full charge, are racing to the exterior of the protein so that they can interact favorably with water. They speak that language of charge. They want to be around water. Those two things simultaneously are the largest contributor of protein folding. That single primary structure of amino acids contains hydrophobic and hydrophilic amino acids inherently. The hydrophobic amino acids try to get to the middle of the protein as it folds. The hydrophilic amino acids try to get to the outside of the protein as it folds, and this is the largest contributor to protein folding. Remember, again, I can't emphasize it enough, there's no such thing as a hydrophobic interaction. The hydrophobic side chains don't care at all about each other. They're simply interacting together or in the same place at the same time because they're trying to get away from water. So here we see a static image of protein folding. I'll show a dynamic one in just a second. But this is the primary structure of this protein. It's the long ribbon of amino acids, nothing really happening. And we fast-forwarded it to the tertiary structure, fully folded. We've got helices and beta strands all kind of glommed together into a fully folded protein. If we were to think about an amino acid that's right there between this alpha helix and this beta sheet here, or beta strand, those would likely be hydrophobic amino acids. They're right in the center. They're excluded from water. They're side by side. Quote, I'm doing air quotes now in my office, air quotes. They're interacting with each other, even though they're really not. They're mutually excluding themselves from water. That's how proteins fold in the most basic sense. It's a little fast, but this is the dynamic image that I was referencing. We can see fully unfolded, or I guess down to the secondary structure because we do see beta sheets. This protein will come together to form a native conformation, and the energy driving that folding is the energy provided by hydrophobic amino acids racing to get away from water and hydrophilic amino acids being content to stay on the outside. Correct protein folding, not surprisingly, is critical for the protein having the right function, because structure equals function. That means correct structure gives correct function, and therefore the viability of the organism, because the organism requires the proteins doing the jobs correctly. Syndromes such as mad cow's disease, Alzheimer's, Parkinson's disease, Huntington's disease, all neurological in nature in these examples, these are all syndromes that at their root are due to proteins being improperly folded. In these cases, proteins unfold partially. Hydrophobic residues are exposed, and those hydrophobic residues race to shield themselves from water. Since these proteins are unfolding simultaneously, rather than folding back up into their native conformations, hydrophobic residues of neighboring proteins race to interact with one another to shield themselves from water. So these amino acids are lining up with each other in order to get away from water. This causes different proteins to aggregate or clump up together and therefore no longer be soluble. It's this clumping up that causes these syndromes. You get these protein plaques in the neurons of the brain, and it gives rise to the syndromes and symptoms of these diseases. As much as the information for proper protein folding is contained in the primary sequence, sometimes that's not always enough. Proteins don't always fold up properly on their own. Sometimes they need a little bit of help. And these helper proteins that aid newly made proteins in folding properly are called chaperones. Chaperone proteins are specialized proteins that allow and help proteins fold into their native conformations. Proteins can also be unfolded and denatured. If you denature a protein, just like we saw for mad cow's disease, you expose those hydrophobic amino acids and they will clump up. You've all seen this with your own eyes. If you've seen a Sunnyside egg be fried, you have witnessed the denaturation and aggregation of proteins. As you fry an egg, the albumin in that egg white unfolds due to the heat, the proteins unfold into their secondary conformations only. They're exposing their hydrophobic residues, and then they race together and aggregate so that those proteins can, those amino acids can be shielded from water. That aggregation, that clumping up, causes that albumin to come out of solution and turn white and be safe to eat as well, because you've killed any bacterial enzymes that might be there also. So heat can denature proteins. These interactions that are keeping proteins into their tertiary shape are largely weak, non-covalent interactions, so heat is enough to break them. Extremes in pH, very, very basic or acidic environments, can also interact and disrupt tertiary structure by breaking electrostatic interactions. Treatments with detergents, you've probably encountered this in some labs you've taken already, or if not, you'll encounter it in the lab you do take for this course. Detergents are amphipathic, so they interact favorably with nonpolar side chains, fooling them into thinking that it's safe to come out into the watery environment. And so in the presence of detergents, proteins literally will act like popcorn, and they'll turn inside out, where the interior parts will come out and the exterior parts will go in. And while heat, extremes of pH, and detergents will all disrupt non-covalent forces, of course, disulfide bridges are always there in our way. Those are covalent bonds. They're very, very strong. And to break those, we need a reducing agent, something like dithiothiorate or beta-mercaptoethanol. Again, compounds you may have encountered in labs you've taken here at Baypath or in other institutions. These reducing agents destroy disulfide bridges by protonating those thiol groups. And so the disruption of the covalent bond here is due to the absorption of protons by those thiol groups. So we'll end, as promised, by talking just for one slide about determining protein structure. We'll keep this very, very brief. This is the experimental techniques that we use to, again, air quotes here, to see proteins in their native conformations. We really can't with our naked eye, but with the help of instrumentation, we can get a sense of what three-dimensional shapes proteins are assuming. The three-dimensional shapes of any protein can be determined, can be measured, kind of visualized, using two primary strategies. One is called X-ray crystallography, and the other is similar to the technology for magnetic resonance imagery in hospitals, MRIs. This is nuclear magnetic resonance, or NMR. Again, I don't want to take too much time to go into severe depth of how these techniques work, but all I would like you to know is that they exist. X-ray crystallography and NMR are two different ways to measure and determine protein shape. For X-ray crystallography, perfect crystals of pure proteins are made, and by shining X-rays into those crystals, the way those X-ray rays are diffracted gives you a sense of the structure of the side chains and the position of the side chains, which allows you to then infer the overall tertiary shape of the protein. For those of you who know Dr. R.C., and many, if not all of you, do, she's the lab instructor as well as our resident biochemist. She has hands-on personal experience with X-ray crystallography, and so if you're interested in this, I encourage you to speak to her. She can give you some cool stories about it, I'm sure. I said that. NMR uses a completely different technology. Here, very, very large and powerful magnets are used to make the hydrogens of the protein vibrate. Those vibrations are then measured by the instrumentation, and how and where those hydrogens vibrate allows the overall position of those hydrogens to be inferred, which then in turn allows you to infer the overall structure of the protein in general. So two very different ways for measuring protein tertiary structure, but both of them equally valid ways to do so, and scientists can get a sense for what proteins look like in three-dimensional shape by using X-ray crystallography or NMR. So to summarize what we talked about here, we've got tertiary structure, which is the fully folded protein, the polypeptide chain fully folded in 3 dimensions. Key, though, that it's a single polypeptide chain. And then we can have quaternary structure. Quaternary structure is the only optional structure. Not every single protein assumes quaternary structure, but any protein that is a complex made up of multiple subunits has quaternary structure. Each subunit is its own single polypeptide chain. We talked quite a bit about the non-covalent forces that hold proteins into their tertiary shape, electrostatic, H-bonds, hydrophobic forces, and of course the covalent force of disulfide bridges as well. Those same proteins, don't forget, also keep subunits together into the full complex of a quaternary structure protein. And we ended the lecture speaking very briefly about protein folding. Essentially, the hydrophobicity of some amino acids causes them to race towards the center. The hydrophilicity of other amino acids make them happy on the exterior of the protein, and that's the primary force driving protein folding. And we ended by speaking very briefly about X-ray crystallography and NMR as two different ways by which we can achieve protein visualization. Said all that. So, as is always the case, thank you for watching. We'll be moving on now to protein function in Lecture 4. If things are starting to get interesting now, we are going to hit a whole new level and gear of fascinating science as we talk about what proteins do and how they do it.